This video is a short note on osmosis. So I'm going to first show you a video where erythrocytes and leukocytes burst when exposed to pure water. So right now, these two types of cells are in an isotonic solution, which means that the concentration of solutes is the same inside and outside. So there is no osmosis. And now we put the cells in distilled water, and you can see that water is flowing inside. Well, you cannot see that, but you can see the cells swelling. And eventually they swell to an extent that the cell membrane cannot resist the pressure and the cells burst open. This is because of osmosis of water from the outside to the inside. This has several implications in biology. One is that when blood is stored, it must be stored in the right concentrations of plasma in hospitals so that blood cells don't swell and burst. Secondly, when drugs are injected into the body, they are not injected in pure water, but the solution must be slightly hypertonic to red blood cells, meaning that the concentration of drug solution is slightly higher than plasma, so that RBCs, when they encounter this drug solution in blood, will shrink slightly, they will not burst. Another implication is the balance between osmosis and electrochemical, or more accurately, electrical gradients in the cells. If you look at image A right here, this is a cell containing different types of solutes: DNA, RNA, protein, and metabolites. It also is surrounded by a yellow colored membrane and has aquaporin water channels sitting in the membrane. Since the concentration of solutes is much higher on the inside and the concentration of water is much higher on the outside, water will automatically flow from regions of high to low concentration and the cell will start to swell and will eventually burst. However, cells also contain other types of channels and pumps. The sodium potassium pump pumps out 3 sodium ions and pumps in 2 potassium ions for 1 molecule of ATP hydrolyzed, which means that when the pump is active, it reduces the amount of cations in the cell, which means that the cell will be negative, electrically negative with respect to the outside. When the cell is electrically negative in the inside with respect to the outside, this transmembrane electrical gradient pushes out chloride ions from the cell through chloride channels. This flow of chloride ions outside the cell results in a higher concentration of chloride ions outside the cell than inside the cell, thus balancing the osmotic pressure. Now this aquaporin molecule is very interesting because it transports there are several different types of aquaporins, but some of them are extremely water selective. They do not transport sodium ions, they do not transport protons, they only transport water. This movie here shows you an aquaporin monomer. It actually exists as a tetramer in the membrane. But this shows you an aquaporin monomer sitting in a lipid membrane which is about 5 nanometers thick, not 3, but 5 nanometers. You see water on both sides of the membrane. The membrane is not shown for clarity. These water molecules are shown in red and white. The white stuff is the protein. This blue here represents charge. We'll come back to that. And the movie you are about to see is the diffusion of a water molecule from this side of the membrane to this side of the membrane. Remember, this is not driven by an osmotic gradient or a pressure. This is pure diffusion. So this water molecule, shown in yellow, will eventually diffuse to this side. This is a molecular dynamics simulation, and we will look into this more later in the course. You can see that there are 2 amino acids which have also been highlighted here. We'll come back to that. And then this water molecule eventually passes on to this side. So at equilibrium, if there are no osmotic gradients, the number of water molecules flowing in this direction and in the other direction will be the same, resulting in no net flow. So until crystal structures became available, we did not know what aquaporin molecules look like. After the crystal structure became available, the key question was what makes it water selective? Why don't protons pass through the aquaporin molecule? And this became more apparent from molecular dynamics simulations. So this image, which is not from a simulation, is a cartoon, shows you this part of the aquaporin molecule. You can see that the direction of the water molecule, the orientation of the water molecules is different in this part and in this part. In this part, the oxygen is facing this NPA region, but in this part, the oxygen is facing the other direction. This change in orientation of water molecules prevents the passing of a proton along these water molecules. So it turns out that the water molecules need to be aligned or need to be free. But in this case, they are not aligned. Oxygen faces one way and then the other way in the two parts of the membrane. So that is one way how protons are excluded. The other way is this macroscopic dipole moment of the two helices, which means that there is a positive potential inside in the middle of the protein, and this prevents the entry of positively charged species. So this movement of water can be captured more clearly in this small video animation. So you can see water molecules entering in one direction and leaving in another direction. So what has this got to do with osmosis? So if one takes this aquaporin molecule and embeds it in some artificial membrane and puts salt water on one side and distilled water on the other side and applies a pressure more than the osmotic pressure from the salty side to the pure water side, then you can potentially purify the salt water into pure water. So this process is called reverse osmosis. It has been known for a long time, and it has been artificial membranes have been used for this. But with the incorporation of aquaporins, the amount of pressure required in order to purify water using reverse osmosis is much smaller. There is a startup in Copenhagen. It's no longer a startup. There was a startup in 2005-6 called Aquaporin that has now started to manufacture these membranes containing recombinant aquaporin molecules, and they are now selling these membranes to several people, including NASA. So this was a short note about aquaporins and the importance of molecular dynamics. And here is an important question. During osmosis, when a cell is placed in pure water, why does water move inside the cells? And you have four options here, and I'll give you half a minute to think about it before I give you the answer. So the correct answer is that the chemical potential of water is higher outside the cells. And that's why water flows from a higher chemical potential to a lower chemical potential. So this is very important to note that a compound, will move from a higher chemical potential to a lower chemical potential, just like a ball moves from high potential energy to low potential energy, or a positive charge moves from a high electrical potential to a low electrical potential. So this was a short note about osmosis, and this concludes this video.